There is a substance that has quietly traveled through human history for more than 3,000 years, used across the Middle East and North Africa, and recorded in some of the earliest known medical traditions. Archaeologists even discovered traces of it inside the tomb of Tutankhamun, where it was preserved among items meant for the afterlife. Ancient physicians prescribed it for problems that ranged from breathing difficulties to digestive disorders and persistent infections that other treatments of the time could not resolve. Across different civilizations, it earned a name that reflected how highly it was valued, the seed of blessing. Now, if you hear something like that, it is easy to dismiss it as folklore or outdated traditional medicine that no longer holds relevance in modern science. In fact, that was my own first reaction when I came across it in the research literature. I assumed it would be one of those historical remedies that sounds impressive in old texts but falls apart under modern biological testing. But the deeper I went into the scientific studies published over the last decade, especially between 2019 and 2024, the more that assumption started to collapse. What actually caught my attention was not the ancient history, but a series of controlled laboratory studies showing something very specific happening at the cellular level. Inside this oil is a compound called thymoquinone. In multiple independent experiments, this compound has demonstrated the ability to selectively trigger cancer cell death in several types of cancer lines, including liver, colon, prostate, lung, and breast cancer cells. What makes this particularly important is not simply that cancer cells were affected, but that healthy cells in the same experimental conditions were largely left untouched. That selectivity is the key distinction in biomedical research. Many substances can kill cells, including both harmful and healthy ones. But a compound that appears to target cancer cells while sparing normal cells is something researchers pay close attention to because it suggests a mechanism that is not just destructive, but intelligent in how it interacts with biological systems. Before going further, I need to be very clear about what this discussion is and what it is not. I am Dr. Becker, and I spend a large portion of my time reviewing peer-reviewed research papers that are not widely discussed outside academic circles. My goal is to separate what is genuinely supported by evidence from what is simply popular or exaggerated online. Everything I share here is strictly for educational purposes. This is not a treatment recommendation for cancer or any other medical condition. If someone is dealing with a diagnosed illness or is currently on medication such as chemotherapy or blood thinners, it is essential to consult a qualified healthcare professional before making any changes. With that said, the scientific evidence surrounding this compound deserves careful attention, especially for those interested in how cellular health changes after the age of 60. Let's begin with the basic protocol that has been used in human safety studies so far, because understanding the framework is important before looking at the mechanisms. The substance in question is black seed oil, derived from the plant Nigella sativa. The active compound we are focusing on is thymoquinone. In controlled human safety trials, the commonly used dose is around 2 teaspoons per day, which is roughly 10 milliliters in total. It is taken with food, and ideally, with a meal that contains some level of dietary fat. This is not a minor detail. Thymoquinone is fat-soluble, which means it is absorbed more effectively when fats are present in the digestive system. Without that, absorption can be significantly reduced, and the compound may not reach optimal levels in the bloodstream. The quality of the oil also matters. The studies and traditional usage both emphasize cold-pressed, organic black seed oil stored in dark glass bottles. Exposure to heat and light can degrade the active compound fairly quickly, reducing its biological activity. This is why it is recommended never to cook with it or expose it to high temperatures. Anything above approximately 150 degrees Fahrenheit can begin to break down the active components. Instead, it is typically used in simple ways: added after cooking onto meals, drizzled over salads, or taken directly by spoon. The cost is relatively low, often around $15 per month, which is one of the reasons it has gained attention in discussions about accessible dietary compounds. Now, beyond the basic usage, the real scientific interest begins when we look at how this compound interacts with the body at the cellular level. There are five primary biological mechanisms that researchers have repeatedly observed in experimental settings. The first of these is what is known as selective apoptosis. Apoptosis is the body's natural process of programmed cell death. Every cell has an internal self-destruct system that allows the body to remove cells that are damaged, malfunctioning, or no longer needed. This system is essential for maintaining healthy tissue. The problem in cancer development is that cancer cells learn how to disable this self-destruct mechanism, allowing them to survive and multiply even when they are abnormal. What the research suggests is that thymoquinone may reactivate this process in cancer cells by influencing key proteins involved in apoptosis pathways. One of the most important of these is caspase 3, which acts like a final execution signal in the cell death process. When activated, it essentially instructs the cell to dismantle itself in a controlled way. In laboratory studies, thymoquinone has been shown to stimulate this caspase 3 pathway specifically in cancer cells. Even more interestingly, in the same experimental conditions, healthy cells did not show the same level of activation, suggesting a level of selectivity that researchers find particularly significant. This is where the conversation starts to move from historical interest to modern biomedical relevance. However, it is important to remember that these findings are primarily based on controlled cell studies, not large-scale human clinical trials. The distinction is critical, and it will continue to come up as we explore the remaining mechanisms. In the next section, we will continue from here and look at how this compound may also influence blood supply formation in tumors and how that connects to broader cancer biology. Continuing from the previous point, one of the most important aspects of cancer development is not just how cancer cells survive, but how they expand and sustain themselves inside the body. A single abnormal cell is not usually the real danger. The real danger begins when that cell learns how to build support systems around itself, especially by creating its own blood supply. This process is known as angiogenesis. In simple terms, it is the formation of new blood vessels. Under normal conditions, angiogenesis is essential for healing wounds and repairing tissues. But in cancer biology, this same process becomes hijacked. Tumors begin releasing chemical signals that instruct the body to grow new blood vessels directly toward them, essentially feeding themselves with oxygen and nutrients that allow them to grow beyond a small size. One of the key signaling molecules involved in this process is vascular endothelial growth factor, often abbreviated as VEGF. This molecule acts like a construction signal, telling the body to build new vascular pathways. In cancer, VEGF becomes overactive, and tumors essentially turn into self-sustaining structures with their own supply network. What the research on thymoquinone suggests is that it may interfere with this process by downregulating VEGF expression. In multiple experimental models reviewed in scientific literature, researchers observed that thymoquinone reduced the signaling activity associated with VEG, in some cases by a significant percentage across different cancer types. A systematic review published in molecular science journals analyzed several independent studies and found a consistent pattern. Across different cancer models, thymoquinone was associated with a reduction in VEGF activity, often estimated between 40 to 60%, depending on the experimental conditions. While these numbers come from controlled laboratory settings, the pattern itself is what matters most in early-stage research, because it shows a repeated biological response across different systems. To understand why this matters, think of a tumor as a growing city. VEGF is like the system that builds roads into that city. Without those roads, the city cannot expand beyond a very limited size. By interfering with VEGF signaling, the growth of the tumor becomes restricted due to lack of oxygen and nutrients. In this state, the tumor is more vulnerable to immune activity and less capable of spreading aggressively. There is also an important comparison here with modern oncology drugs. Some pharmaceutical treatments are specifically designed to block VEGF activity. These anti-angiogenic drugs are used in cancer therapy to slow tumor growth by cutting off their blood supply. While those medications operate at a much higher and more targeted intensity, the biological principle is similar, limiting the tumor's ability to build and maintain its vascular network. What is being studied with thymoquinone is not a replacement for those therapies, but rather a much milder, dietary level influence on the same pathway. This is an important distinction because the potency, dosing, and clinical application are not comparable. However, the overlap in mechanism is what makes the research scientifically interesting. At the same time, there is a practical detail that is often overlooked when people consider compounds like black seed oil. Absorption plays a major role in whether these biological effects can even take place. Since thymoquinone is fat-soluble, it requires dietary fat for proper uptake. Taking it on an empty stomach or without any fat in the meal can significantly reduce how much of the compound actually enters the bloodstream. This is why studies and practical recommendations often emphasize consuming it with foods like eggs, nuts, olive oil, or avocado. Without this step, a large portion of the compound may pass through the digestive system without being fully utilized, which reduces its potential biological impact regardless of its laboratory properties. Now, moving deeper into cellular biology, the second major mechanism involves energy regulation within cells, particularly through the AMPK and mTOR pathways. These two systems act like internal switches that determine whether a cell focuses on growth or repair. AMPK is essentially an energy sensor. When activated, it signals the cell to conserve energy and shift toward maintenance and repair functions. On the other hand, mTOR is a growth regulator. When active, it tells cells to grow, divide, and build new structures. In healthy balance, these two systems work together. But in cancer, mTOR is often overactive, pushing cells into constant growth mode even when it is not appropriate. This uncontrolled activation contributes to rapid tumor expansion. Research published in oncology and molecular biology journals has shown that thymoquinone may activate AMPK while simultaneously suppressing mTOR signaling in certain cancer cell lines. This dual action is particularly important because it essentially shifts the cellular environment away from growth and toward repair. To put it simply, it reduces the signals that tell cancer cells to multiply while increasing the signals that encourage cellular regulation and stability. What makes this even more relevant is that AMPK activation is not exclusive to this compound. It is also activated by fasting, caloric restriction, and regular physical exercise. These are well-established lifestyle factors known to influence metabolic health and, in some studies, cancer risk profiles. The interesting aspect here is that thymoquinone appears to interact with the same pathway through a different biochemical route, which means its effects may complement these lifestyle interventions rather than replace them. There is also an additional layer to this mechanism that becomes particularly relevant for aging populations. AMPK activation is closely linked to autophagy, which is the body's internal recycling system. Autophagy allows cells to break down damaged components, misfolded proteins, and dysfunctional mitochondria. This process is essential for cellular renewal and maintaining tissue health over time. As we age, autophagy tends to decline, which contributes to the accumulation of cellular damage. By supporting AMPK activity, thymoquinone may indirectly support this cleanup process, helping the body remove damaged cellular material more efficiently. This is where the discussion shifts from purely cancer-focused mechanisms to broader aging and cellular health. It is not just about preventing abnormal cell growth, but also about maintaining the internal housekeeping systems that keep cells functioning properly over time. In the next section, we will continue by looking at how this compound interacts with the immune system and how that interaction plays a dual role in both defense and inflammation control. So far, we have looked at how this compound may influence cancer cell survival and blood supply formation, as well as how it interacts with key metabolic pathways like AMPK and mTOR. But there is another layer that is equally important when evaluating any bioactive substance, especially for people over 60, and that is how it interacts with the immune system. The immune system is not just a defense mechanism, it is a highly regulated network that constantly makes decisions about what to attack and what to protect. When functioning correctly, it identifies cancer cells early and removes them before they become a serious problem. But when immune balance is disrupted, either through aging, chronic inflammation, or overactivation, this system becomes less effective or even harmful. This is where the distinction between immune stimulation and immune modulation becomes critical. Many substances on the market claim to boost the immune system. That might sound positive at first, but it is an oversimplified idea. An overactive immune system can be just as dangerous as an underactive one. Conditions such as rheumatoid arthritis, lupus, and Hashimoto's thyroiditis all involve the immune system attacking the body's own tissues. Simply stimulating immune activity without control can worsen these conditions. It is like pressing harder on the accelerator without steering properly. You increase power, but you lose direction. What the research suggests about thymoquinone is not simple stimulation, but modulation. This means it helps regulate immune activity rather than just increasing it. In several experimental and clinical review papers, thymoquinone has been shown to influence both innate and adaptive immune responses in a balanced way. One of the key findings is its effect on natural killer cells. These are specialized immune cells responsible for identifying and destroying abnormal cells, including cancerous or virus-infected cells. Studies indicate that thymoquinone may enhance the activity of these natural killer cells, improving their ability to detect and eliminate harmful cells more efficiently. At the same time, it appears to influence cytotoxic T cells, which are another critical part of the immune system's targeted response. These cells act with more precision, recognizing specific threats and eliminating them without causing widespread damage to surrounding tissue. What makes this combination important is that it strengthens the body's cancer surveillance system without triggering excessive immune aggression. In other words, it supports the parts of the immune system that are responsible for protection while avoiding unnecessary inflammatory overactivation. Alongside this immune balancing effect, another important aspect emerges: inflammation control. Chronic inflammation is now recognized as one of the major underlying factors in many age-related diseases, including cardiovascular disease, neurodegeneration, and certain types of cancer. It is not the short-term inflammation that occurs during injury or infection that is problematic, but the long-term, low-grade inflammation that persists in the background over years. Research has shown that thymoquinone may reduce the production of key inflammatory cytokines, including TNF-alpha and interleukin-6. These molecules act as chemical messengers in the immune system, and when overproduced, they contribute to a state of chronic inflammation. By reducing these signals, thymoquinone may help shift the body away from a constant inflammatory state and toward a more balanced immune environment. This does not mean suppressing the immune system. It means reducing unnecessary background noise so that immune responses can function more precisely when needed. This dual effect, enhancing targeted immune activity while reducing chronic inflammation, is one of the reasons this compound has been studied not only in cancer-related research but also in conditions like gastrointestinal infections and metabolic disorders. For example, one study published in a Biological Sciences journal examined the effects of Nigella sativa in combination with standard therapy for Helicobacter pylori infection. This bacterium is known for causing chronic stomach ulcers and is classified as a significant risk factor for gastric cancer if left untreated. In that study, black seed oil demonstrated comparable effectiveness to triple antibiotic therapy in reducing bacterial presence, but with a notable difference. The antibiotic treatment tended to disrupt the gut microbiome broadly, affecting both harmful and beneficial bacteria. In contrast, black seed oil showed a more selective action, targeting the harmful bacteria while preserving much of the beneficial microbial environment. This is significant because gut health plays a major role in immune regulation. A disrupted microbiome can contribute to inflammation and weaken immune efficiency over time. Maintaining microbial balance is therefore not just about digestion, but about long-term immune stability. When you connect these findings together, a broader picture begins to form. The immune-modulating effects of thymoquinone are not isolated actions. They are part of a wider system that influences inflammation, microbial balance, and immune surveillance simultaneously. It is also important to consider how this interacts with aging. After the age of 60, the immune system naturally undergoes a process called immunosenescence, where its efficiency declines. At the same time, chronic inflammation often increases. a phenomenon sometimes referred to as inflammaging. This combination creates a biological environment where disease risk increases while repair capacity decreases. Within this context, a compound that supports immune precision while reducing unnecessary inflammation becomes particularly relevant, not as a cure, but as a potential supportive factor in maintaining balance. In the next section, we will move into one of the most fundamental mechanisms of all, how this compound may influence DNA protection and cellular damage repair, which forms the foundation of long-term cancer risk.